Good morning, everyone. Today, my presentation will focus on Mycobacterium tuberculosis, one of the most important and prevalent bacterial infections in the world. I'll discuss its habitat, morphology, cultural characteristics, biochemical characteristics, host range, virulence factors, antibiotic resistance mechanisms, and epidemiology. Let's get started. Tuberculosis is an infectious bacterial species responsible for tuberculosis, a potentially life-threatening lung disease. Transmission of TB occurs through the air from someone with an active infection in their lungs or throat when they speak, sneeze, or cough. Treatment of this bacterium has been challenging due to the rise of drug-resistant strains. Proper diagnosis and prevention of TB are critical for successful global public health initiatives. Tuberculosis is a pathogen that primarily affects humans, but can also infect other animals. It is usually transmitted through the air when an infected person coughs or sneezes, and is common among cattle and non-human primates, causing respiratory infections. Its transmission is highly contagious and can spread rapidly. Tuberculosis is an rod-shaped bacterium with a cell wall structure containing mycolic acids, a complex mix of long-chain fatty acids, which provide a high level of antibiotic resistance. These fatty acids are difficult to penetrate and destroy, resulting in a high level of antibiotic resistance compared to most other bacteria. Slide 5 of this presentation examines the cultural characteristics of Mycobacterium tuberculosis, the culpable bacterium for tuberculosis. This species of bacterium usually has a slow growth rate on solid media, with colonies materializing after 3 to 6 weeks. In contrast, liquid media gives a faster result. Tuberculosis is a bacterium primarily found in humans and other animals that requires oxygen to survive. It is distinguishable under the microscope through its unique acid-fast staining characteristics, as it is able to retain certain stains when exposed to acidic solutions. Tuberculosis is a human pathogen with the potential to cause tuberculosis, a serious infectious disease. It is not exclusive to humans, being able to infect other animals, which is why it is vital to identify and reduce contact with the bacterium to protect both individuals and animals. Tuberculosis has a range of virulence factors that make it possible to exist in the human body. These include the lipids found in its cell wall, proteins, and enzymes that modify how the human body responds. These components interact to elude the immune system, causing prolonged illnesses. These can cause serious problems for fragile populations. Tuberculosis can demonstrate antibiotic resistance caused by its cell wall structure and mutations in genes responsible for drug metabolism or transport. These mutated genes can cause proteins to break down or expel antibiotics from the bacterial cell, or reduce the energy needed for bacterial growth. These mechanisms of antibiotic resistance can create difficulties in treating this bacterium, as some antibiotics may be rendered ineffective. Tuberculosis, or TB, is a serious and highly contagious disease. Estimated at 10 million people infected annually, the number of fatalities from TB is an alarming 1.4 million. Developing countries suffer the most from this health crisis, yet it is also a serious problem for certain developed countries' vulnerable populations, such as immigrants and the homeless. Fortunately, there are treatments available to help combat it. Tuberculosis is a pathogen that primarily affects the lungs, causing coughing, chest pain, and difficulty breathing. It can also spread to other parts of the body, including the brain, bones, and kidneys, with varying degrees of severity. To curb the growing threat of this pathogen, it is essential to understand the factors believed to lead to its transmission. Tuberculosis is an infectious pathogen responsible for a large number of fatalities annually. Its infectious nature is attributed to its capacity to circumvent the host's immune system. Through intricate immunological reactions, Tuberculosis can thrive in a diverse range of hosts without being utterly abolished. Investigating these responses may be instrumental in uncovering novel treatments and potentially salvaging lives. A combination of approaches, including chest X-rays, sputum smear microscopy, bacterial culture, nucleic acid amplification tests, and rapid diagnostic tests, are typically used to diagnose TB. Together, these tests can provide an accurate diagnosis. Proper ventilation and isolation of infected individuals, as well as vaccination, are significant measures to prevent the transmission of tuberculosis. Vaccines may vary in their efficacy depending on the population being vaccinated, but when combined can be very effective. Mycobacterium tuberculosis is the bacterium responsible for causing tuberculosis, a serious and highly contagious condition. Treatment requires a long-term regimen of antibiotics, usually in combination, taken over a period of 6 to 9 months or longer. Selection of the specific antibiotics used depends on the strain of M. tuberculosis present, as well as the patient's individual characteristics. Completing the entire prescribed treatment plan is essential to prevent the bacteria from becoming resistant to the medication. Mycobacterium tuberculosis is a dangerous infectious disease that must be taken seriously. Effective diagnosis and treatment is essential in controlling the spread of tuberculosis, as is preventing transmission and increasing vaccination rates. This presentation has hopefully provided you with a greater understanding of this virus, and your attention is appreciated.